Hello everyone, my name is Uzair Hashmi and for my profile details, please visit sqadia.com. So let's start this particular chapter. In this lecture, we are going to discuss about the compartments of different body fluids. We are going to discuss in details what are the different types of compartments. We are going to discuss each and every single compartment in details and along its regulation, and how these compartments of the body fluid play a very critical role, in maintenance of the homeostasis and what happens under the abnormal physiological conditions. So in this lecture, we'll start off with the introduction to the compartment functions. We'll move towards what are the daily requirements for the fluid intake, and how this particular intake is managed by the loss of the fluid. We're going to discuss what are the different types of the fluid compartments and the body fluid percentages in both newborns, adults, and old women. After that, we are going to discuss about different constituents of the extracellular and the intracellular fluid. We're going to discuss the osmotic balance in the extracellular and the intracellular environment, and we are going to discuss under what conditions, for example, there is what happens in the case of hypotonic environment, hypertonic, and the isotonic environment, and what are the changes that occur during these particular conditions. We're going to discuss what are the conditions which will give rise to hyponatremia or hypernatremia, and after that we are going to discuss what are the types and the causes of extracellular edema. After that, we are going to discuss what happens when the edema is caused by the heart failure, and what happens when the fluid in the potential spaces of the body, they don't keep their volume intact. So let's get started. So this chapter is all about the regulation of the body fluid volume. We'll discuss how if you increase the intake of the water, how that particular intake is managed by the kidneys. We are going to discuss what are the constituents of the extracellular fluid, what are the different types, including the cerebrospinal fluid, interstitial fluid, transcellular fluid. We are going to discuss in the coming slides. We are going to discuss how this particular body fluid maintains the acid-base balance, and what are the different functions that control the fluid exchange between the extracellular and the intracellular compartments. So it must be known to you that the daily requirement for the intake of water is about 2300 mLs a day. That is about 2.32 to 3 liters a day, and that is the case of a stagnant position when there is very lack of physical activities, sedentary lifestyle. So out of this particular volume, 2100 ml of the water is being intake in terms of the body fluids when we intake water in the form of liquid. The rest of the 200 mLs per day, that is being synthesized as a result of metabolic reactions from primarily the oxidations of the carbohydrates. That basically adds to 200, making up to 2300 ml per day of the daily fluid requirements. So we discussed the intake of the water. Now let's discuss the daily loss of body water, and it occurs through different mechanisms. The number one, which is the insensible water loss, plays a very critical function, because it is a type of the water loss that is continuously occurring without our conscious recognition, because this particular type of the water loss occurs through respiration, that basically from evaporation through respiration through the respiratory tract, and this also occurs through the skin. Now it accounts for around 700 ml of the day, and this particular thing, insensible water loss, cannot be precisely regulated. However, there are different conditions under which these can be minimized, and under these conditions, for example, take an example of the skin. There is a particular layer, as mentioned here in this particular slide, that is cholesterol-filled cornified layer of the skin. In this particular type of the layer, this basically, and the cholesterol, since is a type which is hydrophobic in nature, so it will act as a water repelling barrier. So this would help to resist the evaporation through the skin. So this plays a very critical function in minimizing the loss of water through the skin. So insensible water loss will also occur under such individuals which do not have any kind of sweat glands. The other category of the daily loss of body water includes the water loss through the sweat. Now, it varies greatly, and it depends upon two things. Number one is the physical activity, how much physical activity you're putting in your daily life, whether you are sedentary in your lifestyle or you are working in the office hours 9 to 5 without having any kind of exercise. Contrary to that, you have a very active physical day. So normally there is about 100 mLs per day loss through the sweat, but that increases in the very hot weather situations where in case of the heavy exercise it increases up to 1 to 2 liters a day. And that of course will activate the thirst mechanism where you will be receiving the need to quench your thirst, so that basically it balances off the loss of the fluid. The other thing is the loss of water through feces. Now it is also 100 ml per day, but it can significantly increase to even liters when there is a severe form of the diarrhea, and that is why ORS is being given to prevent or to balance the loss of the electrolytes from the body. So the daily loss of body water, it can also occur through the kidney, and the kidney plays the most important role to balance the water intake and the electrolyte intake along with the output. So urine volume, it varies dependently, and that is the tremendous amount of that is the tremendous potential for the kidneys to regulate the urine volume from as low as 0.5 liters a day to about 20 liters a day, and that entirely depends upon the amount of volume or the amount of fluid you are intaking. Now, the kidney, of course, will be adjusting to the excretion rates that will match precisely to the rate of intake of the fluid. And of course, this is also followed by the electrolytes. So kidney plays, a key role in regulating the body fluid, and majorly the extracellular fluid. So to summarize the daily intake and output of the water in milliliters a day, you intake a total of 2300 mLs, that is about 2.3 liters. And that out of 2.3 liters, 2.1 is being ingested in the form of fluid, and the rest 200 mLs a day is being produced as a result of metabolism. Now, what are the different mechanisms or the pathways through which the body fluid or the body water is being lost includes, of course, the insensible, that is through skin, that is about 350, insensible through lungs, because the insensible water loss, as I have discussed, occurs through two basic processes, two basic pathways for that. Number one is the skin, number two is the lungs through the respiratory tract. The other includes the sweat, which is about 100 mLs a day, and includes the feces, which is also 100 mLs a day. And the urine, if we take an average of from 0.5 to even 4 liters, there is an average excretion of about 1.4 liters of the urine under normal physiological conditions, causing the output that exactly or precisely equal to the fluid intake, that is 2300 a day. Over here, under the prolonged or the heavy exercise, fluid ingested is entirely dependent upon the type of physical activity that you're doing. So it will, of course, dependent upon how much exertion you're putting, that will, of course, ultimately increase the thirst mechanism, which will cause you to drink a tremendous amount of water. So, of course, it will, of course, alter the total intake as well. So in case of the prolonged heavy exercise, the total output would be about around 6.6 liters a day. So I've got two questions for you. Number one is why burn patients are given more fluid And number second is that what are the dry, why there is a dry feeling in the respiratory passage when there is a cold weather. So I want you to pause this video and think about for a while about these two questions, and then I'll discuss the answers. So uh, let's start off with the very first question: why burn patients are given more fluid? So what happens, we have to understand, whenever you're going to answer a particular question, the very first thing that you should keep in your mind is to understand what the question is asking. 90% of the time you know the answer, but you don't know how to cater that particular question, how to dissect that particular question to really understand what the question is being asked. So let's start off with this: why burn patients are given much more fluid. So burn patients are the patients with the burn, and what happens when there is a burn? There is several different categories of burn, including the partial thickness burn, first degree, second degree, third degree burn. Second degree is known as the partial thickness burn. So whenever there is a situation of where a person is being victimized with burn, the skin layer would be eradicated or denuded. So in this particular category, when there is a case of a burn victim, the skin layer would be damaged or denuded. This would of course decrease the cornified layer of the skin, which will be containing the cholesterol, right? So that particular barrier, which was resisting the loss of water, has been removed or has been minimized. There is very less amount of that particular cornified layer that is present. So the water loss increases tremendously. The water loss increases up to tenfold in some circumstances. For example, if the water loss under the normal physiological conditions were around 300 ml a day, it will increase to about 3 liters to 5 liters a day, and that would severely deprive the body with the water. So that is why the burn patients are given much more fluid, and usually they are given much more fluid through the intravenous route, to tackle the loss of water through in that particular scenario. Moving towards the second question: why there is a dry feeling in the respiratory passage in the cold weather? So to answer this particular question, you should know that what happens in the summer. So what happens when you inhale the air becomes saturated with the water droplets, and it increases the atmospheric pressure to about 47 millimeter mercury. Now this pressure outside the body, when there is air which is to be inhaled, which has not been inhaled yet, is less than 47 millimeter per mercury, right? So this would increase it would create basically the water pressure or vapor pressure gradient from inside of the body to outside of the body. So there is a continuous loss of water through that particular process, right? So moving on, what happens in the cold weather? When there is a cold weather, the outside of the vapor pressure further decreases to nearly 0 millimeters of mercury. So now the gradient that was established during the summer was less as compared to now, because inside there is a saturation of about 47 millimeters of mercury, and outside is nearly 0 millimeters of mercury, right? So what would happen? In this particular scenario, there would be tremendous amount of the water loss in this particular scenario, because the vapor pressure gradient is much more, the difference in the vapor pressure is much more, so there would be tremendous loss of water in terms of the winter. So in this particular case, that respiratory passages would be felt dry. So that is why there is a dry feeling in the respiratory passage in cold weather. So you must be aware when the winter is coming. So moving on towards the body fluid compartments. Of course, it is divided into two major portions: the extracellular fluid and the intracellular fluid. So let's start off with the extracellular fluid. It includes the interstitial fluid, and the interstitial fluid is a type of fluid in which the tissues of a particular organ, they bathe. Furthermore, it includes the blood plasma. It includes the transcellular fluid, which is around 1 to 2 liters. The transcellular fluid, includes a variety of different types of fluids. Number one is the cerebrospinal fluid, the CSF. The number one is the intraocular, which is the fluid of the eye. The third one is the pleural fluid. This is followed by the peritoneal fluid, which is the fluid of the abdomen. Pleural is for the lungs, of course. We are going to discuss when we have a figure of the pleural cavity, of course. Same goes for synovial and the digestive secretions. So, to summarize, a 70 kg adult, the total body water is about 60% of the body, and that is about 42 liters. In terms of the intracellular compartment, the rest of the 28 of the 42 liters of the body fluids are inside the cells, and a body contains about 60 to 75 trillion cells. So they collectively are called the intracellular fluid. So if we have a look on this particular figure over here, so there is an extracellular fluid of about 14 liters. The intracellular fluid is about 28 liters. The output, you have to intake the body, and the output is through kidneys, excretion through kidneys, lungs, and feces and sweat and skin, including both the insensible water loss through sweat, water loss through feces, and kidney maintaining the osmotic potential. Right. So moving on, that is how a body fluid percentages over the age are changed. So the newborn infant would be containing about 80% of the body fluid, as compared to the adult male, which will be containing about 60%, and the adult female, which will be containing about 50% of the total body. So what are the constituents of the extracellular and the intracellular fluid? So the ionic composition, it's first, it's important to consider that the ionic composition of the plasma and the interstitial fluid is similar. It must be known to you. The second thing is, you must be known that there is a slightly high concentration of the positive ions in the extracellular fluid, and there is a slightly negative concentrations of the negative ions in the intracellular fluids. So, however, the overall, if we take the whole picture, the bigger picture of extracellular and the intracellular fluid, the net charge on the body is 0. So, the cations, if we take an example of the cations, sodium, of course, sodium ions is present in the abundant quantity in the extracellular fluid as compared to the intracellular fluid. The potassium, contrary, is present in the abundant quantities in the intracellular fluid as compared to the extracellular fluid. Same goes for calcium and magnesium. Calcium is present outside and magnesium inside. Much of the magnesium is present inside, and much of the calcium is present inside. So, talking about the anions, chloride ions, of course, is present outside the cell in the extracellular fluid. This is followed by the phosphate ion, which is present in the abundant quantities in the intracellular fluid. Bicarbonate ions are present outside in the extracellular fluid, and the protein, much of the protein, of course, is present within the cells, so that is containing a bigger chunk of the intracellular fluid compartment. Now, this graph over here is representing the non-electrolytes that are present in the plasma. Right, so the major percentage is occupied by the lipids or the fats, including the phospholipids, which is about 280 milligram per deciliter. This is followed by the concentration of the cholesterol in the plasma, the neutral fat, and then comes the glucose, which is being discussed so much. This is followed by the urea, the lactic acid, uric acid, the creatinine, which is the breakdown product of the muscle creatine, bilirubin, the breakdown of the red blood cells, bile salts. They are present in very trace amounts. So let's have a look on an in-depth overview, an in-depth discussion about the different types of salts and their concentrations in the plasma, in the interstitial and the intracellular fluid. So this was to give you an overview, and this would discuss a more in-depth details. Similar, as mentioned in the previous slide, the potassium is much more in the interstitial as compared to the intracellular. The potassium, contrary, is much more in the intracellular as compared to the interstitial fluid. This is followed by the calcium, of course, as well, calcium and magnesium as well. The calcium is present abundantly in the interstitial as compared to intracellular, and the magnesium, of course, is present much more in the intracellular as compared to the interstitial, as shown in this particular figure. Potassium is present much more inside as compared to outside. Similarly, sodium is present much more outside as compared to inside. Same goes for chlorine, bicarbonate, proteins, and phosphate as well. And all of these has been mentioned over here. So moving on to the basic principles of the osmosis and the osmotic pressure. So let's start off with the definition of osmosis. So what do you think in osmosis is? Osmosis is basically the net diffusion of water. So in osmosis, the water will be moving. And in which particular direction water will be moving? The water will be moving from a region of low water potential to a region of high water potential. So it means that water will move through the selective permeable membrane, where there is from a region of low solute potential to a region of high solute potential. So we are going to discuss in details in the coming slide. Over here you can see the initial stage. Here is the pressure P1A, here is the pressure P2A. Over here the concentration of solute is much more as compared to here, as can be evidenced from the red dots that basically represents the dissolved salts. So this selectively permeable membrane will only allow the water to move from one compartment to another compartment without the movement of the ions or the dissolved salts. So with the passage of time, after a particular time, there would be a final state where the water will have moved from a region of low solute potential to a region of high solute potential, increasing the water potential or increasing the osmotic pressure over here, which can be represented and calculated by the difference of the height of these two compartments. So water has moved from a region of low solute potential, where there was less concentration of solute, to a high solute potential. So that is why you can see the water outside the water lemon, when you have, sprinkled salt over it. Yeah, because there is abundant quantity of water in the water lemon. So what happens is that watermelon, so what happens in that is that when you have sprinkled salt over that, so the water tends to move from a region of low solute potential to a region of high solute potential, and you can see the water oozing out of the watermelon in that particular circumstance. So what are the relations between the moles and the osmoles? It is one of the important slides of this particular lecture, in that you must be able to, distinguish between the moles and the osmoles. There's very slight difference, and I hope you will be able to understand and explain better after this particular slide what is the difference between the moles and the osmoles. So the total, of course, you are familiar with the moles by now. You must have been. The total number of the particles in a solution is measured in osmoles. The total, I repeat, total number of the particles in a solution is measured in osmoles. And one osmole is equal to 1 mole of the solute particles. Of course, 1 mole, of course, is equals to the Avogadro's number. So, but here's the important difference. What you have a particular molecule, and that particular molecule, when you dissolve in a solution, let's take an example of a sodium chloride. So you have 1 mole of the sodium chloride with you, and its osmolarity is 1 osmole as well. So what happened when you dissolve that particular 1 mole of the sodium chloride into the water? It dissociated into sodium ions and the chloride ions. So now there are two kinds of ions that are floating, right? So one is the sodium ions and one is the chloride ions. So the number of moles for the sodium chlorides remain the same, that is 1 mole. But here is an important difference. If you are to determine the osmolar concentration, it would be, of course, 2 osmoles per liter. So a solution containing 1 mole per liter of a solute, when dissolved in the water and it dissociates into two active particles, the osmoles, so the osmolarity increased from 1 osmoles per liter to 2 osmoles per liter, right? So that is similarly, if you have a particular compound or a molecule, let's take an example of the sodium sulfate, which when decomposed in the water would produce three active particles. So 1 mole per liter molarity of the sodium sulfate will be changed into 3 osmoles per liter, because the osmose, when we talk about osmose, it is basically calculated by the presence of number of active particles. So it basically refers to the number of active or osmotically active particles that are present into the solution, rather than, of course, their molar concentration. There is a relationship between moles and osmoles, and you have been told in the previous slide that when you talk about moles, it is the total number of particles in a solution, right? So moles is a total number of particles in a solution. That is, of course, the Avogadro's number. And when we talk about osmoles is the total number of osmotically active particles in the solutions. So molarity, of course, as you know, is the moles of solute divided by total in terms of liters of solutions. So in general, osmose is much larger term for expressing the osmotic activity, and you can't explain the the activity in terms of osmose, because it would be around 0.0003 osmose, let's say. So the term milliosmose has been coined to which of course equals to 1/1000 parts of the osmose, which is of course commonly used. So osmolality is basically when the concentration is measured in osmoles per kg of water. And osmolarity is a term which is coined when you calculate the concentration in terms of osmoles per liter of the solution. So osmotic pressure, of course, must be the precise amount of water that is required to prevent the osmosis or the movement of the water from one point to another point. So let's say if I am pushing you with a force of 4 Newton, right? So you have to exert at least 4 Newton of the force to prevent being pushed back. So similarly, osmotic pressure is the pressure that is required to prevent the osmosis. So if the water is moving from, let's say, from here to here, and the osmotic pressure is about let's say 5, in order to provide the similar osmotic pressure, you have to provide an external pressure of 5 so that to prevent the movement of water from here to here. So of course, the higher the osmotic pressure of a solution would be, it means that there is a low concentration of water and there is a high concentration of solute. So the water has to move no matter what, unless you have to apply a stronger force to prevent the movement of water. So up till now we discussed the introduction, we discussed the daily intake and outtake, we discussed body fluid percentages in newborn, man and woman, we discussed the osmolar substances in extracellular and the intracellular fluid, and we discussed the basic principles of osmosis and osmotic pressure. So we discussed in the very last slide what is the relationship between moles and osmoles, and now we are moving onwards with the difference of the osmotic environments. So there are 3 kinds of osmotic environments, either hypertonic, isotonic or hypotonic. So isotonic environment is a kind of environment where the solute and the water potential inside the cell, inside the compartment and outside the compartment is equal. In terms of hypotonic environment, the water potential, the solute potential inside the cell is much more as compared to the outside. So water tends to move from outside or the extracellular fluid into the cells, making the cells look bulky, as shown over here. You can see, because there is a movement of water inside, so the environment at that particular situation would be known as the hypotonic. Same goes for the hypertonic environment. Now, over here, hypertonic environment means that the solute potential, the concentration of solute in the extracellular fluid outside the compartment is much more as compared to inside the compartment. So water tends to move from where to where? Yeah, from inside of the cell to outside of the cell. So what happens over there is that when the water will be moving from inside to the outside, the cell would shrink in size as shown over here. The water will move outside of the cell. In case of hypertonic, it will be same in terms of isotonic, and water will be moving inside in terms of the hypotonic environment. So what are the different osmolar concentrations? This is another figure to explain the isotonic, hypertonic and the hypotonic environment. In this particular figure, isotonic, the concentration of solute inside and outside of the body remains the same. In case of the hypertonic environment, here you can see that the number of dots outside in the solution is much more than inside. So the water tends to move from inside of the cell to outside of the cell. So what it means is that the cell will shrink in size as shown in the figure over here. Now let's talk about the hypotonic environment. In contrary to the hypertonic environment, what is happening over here is entirely opposite of what is being happened in the hypertonic environment. So in case of hypotonic environment, as you can see, the concentration of solute inside the cell is much more as compared to outside of the cell. So the water tends to move from a region of low solute potential to a region of high solute potential. So in this particular case, the water will be moving from the solution into the cell. So it will cause the cell to increase in their size, the cell to swell up. So these are the three basic different types of the osmolar environment that exists in the body. So what happens when there is a fluid shift and the osmolarity? In this particular figure, we are going to discuss under what conditions if you add something. So how will the volume of the intracellular and the extracellular fluid would change, and how it would change the osmolarity as well. So under normal state, as you can see, the intracellular fluid is much more as compared to the extracellular fluid. So in this particular case, let's discuss this particular case. Here, isotonic NaCl solution has been added. Of course, it is being added to the extracellular fluid. Since the osmolarity is same, it means that there is no solute potential difference in the isotonic environment. So it will only increase the volumes in terms of liter of the extracellular fluid without altering either the osmolarity of the intracellular or the extracellular fluid or the volumes of the intracellular fluid. It will only increase the volume of the extracellular fluid because you have added the NaCl solution that is isotonic with the established conditions and the normal physiologic conditions. Now let's discuss B, when you added a hypertonic NaCl solution. So you are adding a hypertonic NaCl solution. So what will happen in this particular case? You are adding abundant amount of solutes in the extracellular fluid, right? So yeah, the number one thing is that because it's a solution as well, this would contain the extracellular fluid will increase in terms of volume because of two things. One, of course, because the solute potential outside or the extracellular fluid has been increased as compared to the inside. So the water, if you remember, through osmosis will tend to move from a region of low solute potential, that is from inside, to a region of high osmotic potential, and that is the extracellular fluid, because you added hypertonic NaCl solution. Now it will, shift the water volume from inside to the outside, and the water will increase. That is why this particular compartment will shrink and this particular compartment will increase. Now what happened, because the solute had been added in the extracellular environment, so the osmolarity of the extracellular environment will increase. And because the water shifted from inside to the outside, so the remaining concentration of the solute increases, hence it will also increase the osmolarity of the intracellular fluid as well. And that is why the osmolarity in both compartments would increase, and the volume of the intracellular compartment will decrease, but the volume of extracellular compartment will be increased in this particular case. Now let's take an example of part C, where you have added the hypotonic NaCl solution. In this particular type, of course, what you are adding abundant amount of water. right? You're adding abundant amount of water with a very slow with a very small concentration of NaCl. So you are adding over here because you are disturbing the equilibrium. So what would happen, think of an example that you added you made a tea which was of course very sweet. So the only solution you had is to add, you know, much more milk in it, so that the sweetness, the constant amount of sweetness decreases to a level where, you know, you can drink it. So similarly, when you add, you will add hypotonic, so that milk was the simile of the hypotonic environment that you added to, you know, lower the concentration of glucose, or the lower the concentration or the osmolarity of glucose in that cup of tea. Similarly, over here, as you can see, when you will add a hypotonic solution of the NaCl, the osmolarity in it will decrease, of course, because you added abundant amount of water in the extracellular fluid. So the number one thing is the decrease in the osmolarity of both these compartments, and when the water has been added, so in comparison to the extracellular fluid, there is much more increase in the volume of the intracellular compartment. And why is that? I want you to stop this video and think for a second, and then we'll resume. So in this particular example, you can see that you have added a hypotonic environment of the sodium chloride. So it will, you know, increase the solute potential inside. So the water tends to move from the region of low solute potential to region of high solute potential, as we have been discussing in the previous slides as well. So this would cause the movement, a net movement of the water from outside to the inside. It would of course increase the cell volume, of course, the intracellular compartment, and which has been seen by increase in the dotted line of the intracellular and both the extracellular compartment. So under what circumstances hyponatremia and the hypernatremia is being occurring? So there are 4 different conditions. The first 2 are discussing the hyponatremia cases, and the last 2 are discussing the hypernatremia cases. So let's take an example when there is adrenal insufficiency and the overuse of diuretics. So under that particular condition, the extracellular volume will be decreased because the diuretics are being overused, so there would be abundant loss of water, and the intracellular fluid volume will of course increase. Take an example of hyposmotic overdehydration. This would occur in case of the excess antidiuretic hormone is being secreted, or the bronchochronic tumor. In both of these situations, the plasma concentration of the sodium will decrease, but over here the extracellular fluid volume will decrease because there is an excessive ADH, antidiuretic hormone, which is also known as the vasopressin, which of course retains the water because it's being stimulated or activated under the conditions where there is less amount of water that is present in the body. So under these circumstances, when excess ADH is being secreted, water of course is present under the normal circumstances, but what will happen in that particular case would be retention of water, which would of course, in terms of the ratio of volume to solute, the concentration of sodium in the plasma would of course decrease. Now, what are the conditions for the hypernatremia? This means that under what circumstances the plasma concentration of sodium would increase. So take an example of the diabetes insipidus or excessive sweating. So what happens in that particular case, the body is producing abundant amount of there is an abundant amount of fluid loss in both of these cases, in case of excessive sweating, in case of diabetes insipidus. So the fluid loss is much more. This would, the remaining fluid will be of course containing the high amount of the sodium. This means that the extracellular fluid volume will decrease because there is abundant excretion of water either through urine in terms of diabetes insipidus, or either through the excessive sweating. And of course this would ultimately result in the decrease in the intracellular fluid volume. What happens in the Cushing disease or the primary aldosteronism, which is known as the Crohn's disease as well. So in these particular two examples, the aldosterone is produced in an excessive quantity, which of course will decrease the renin production. So this would of course increase, because the renin is being produced when extracellular volume has to be decreased, but excessive aldosterone will suppress the renin system, which will of course suppress the whole renin-angiotensin pathway for the regulation of the extracellular fluid volume. So this would result in the increase in the extracellular fluid volume. When it will be increased, of course, this will cause the decrease in the intracellular fluid volume because of that. So extracellular edema. Of course, it's an edema, the inflammation or the swelling because of the accumulation of the water in the regions that is outside the cell. That is why the name given, the extracellular edema. Of course, in this particular scenario, there is an excessive fluid accumulation in the extracellular spaces. When we talk about the causes, there can be abundant amount of causes for the edema. Of course, the most important and the primarily is the leakage of the fluid from the plasma that is present in the blood from the blood plasma into the interstitial fluid of the capillaries. The other important reason is that it can be recovered, but under certain circumstances, when there is a failure of the lymphatics to return the body fluids that has been entered into the plasma or into the capillaries to back into the blood, under certain circumstances, there would be accumulation of the blood. There would be accumulation of the interstitial fluid. So this would slowly and steadily give rise to the condition of the edema. Many other reasons includes, of course, the lymph blockage, or when there is a blockage of the lymph, the fluid won't be able to move back into the plasma, and hence would cause inflammation, and slowly and steadily edema will of course be raised. The plasma protein plays a very important functions, because plasma proteins usually they don't leak out into the interstitial, but under such circumstances, when the capillary permeability or the filtration increases, in such circumstances, because the capillary permeability has been increased, for example, if the permeability was this and the protein size was this much, so proteins were not able to move from this particular path. But what happens when the permeability is increased between the capillaries, that is how the capillary would increase in the permeability. So the proteins which were initially couldn't pass through the capillaries into the interstitial are now able to move past through the capillaries. So the protein plasma protein in the such circumstance would leak out, and of course they would have no way to come inside back. Now this would of course because proteins are a type of solute, so it would of course cause much more movement of the water into that particular portion where the protein has been leaked, so it would of course cause the edema to occur. There is of course the blockage due to infections, which includes one of which is the filarial nematodes. In this particular example, this particular nematode or worm, it basically blocks the lymphatic system. So the same thing that is happening in the lymphatic blockage of the edema will of course be occurring during this particular infections, because they are blocking the lymphatic system. The other important function is the blockage due to cancer, which will of course cause the obstruction primarily in the portal venous system. If there is an inflammation or the blockage, the body fluid will of course be summoning up or collecting into the peritoneal cavity, giving rise to a condition known as ascites. So the causes of the extracellular edema, number one is the increased capillary pressure. This would of course be caused by excessive kidney. This would of course result in the excessive kidney salt and the water retention. When this would be occurring, there would be chances that there is a chronic and acute renal failure. The other example for the increased capillary pressure there is a high venous pressure and high venous constriction, because when the venous capillaries or the venous veins or the veins, they are contracted, they are constricted under the action of different hormones, the pressure would build up. when the pressure would build up, there is reason that there is a strong evidence that heart failure can occur owing to the venous obstruction, because the venous constriction led towards the myocardial infarction or the heart failures. This would of course lead to the failure of the venous pump. When venous pumps would fail, of course this would cause abundant of things, including the muscle paralysis would occur, because there would be no oxygen to be supplied, so there would be no contraction, hence leading to the muscle paralysis. Immobilization of the part of body would result as a result of muscle paralysis, and of course the venous walls will fail. This means that the walls which used to prevent the two-dimensional flow of the blood would of course be failed, so now the blood will move from in both direction under the absence of the complete regulation of the pressure. This would of course will decrease the atrial resistance. Excessive body heat will be produced in this particular point, because the atrioles are not being constricted, because one of the very important mechanism for the atrioles is that they resist the loss of heat. So when there is loss of atrial resistance, body heat will be produced in abundant amount of quantity, and there would be of course insufficiency in the sympathetic system, sympathetic nervous system, because of this decreased atrial resistance. And of course the vasodilator drugs of course further would cause the the dilation of the arteries, where the excessive body heat will be produced, and hence it would make the condition much more severe. Now let's discuss the decreased plasma protein concentration, where the loss of protein occurs in terms when there is some problem associated with the kidney. Usually occurs in the nephrotic syndrome. There is abundant loss of proteins. And loss of protein can also occur through the denuded skin areas, which we discussed in the previous slides as well, where I asked you a particular question about why burn patients are being given excessive fluids. This occurs in terms of burns or wounds where there is a loss of protein from the denuded skin area, so decreased plasma proteins hence. It of course also leads, a decreased plasma protein can also occur because of the failure to produce proteins, for example in terms of the liver disease, for example cirrhosis, or if there is a serious protein deficiency, or calorie malnutrition. It also occurs the edema because of increased capillary permeability. Now, when there is increase in the permeability of the capillaries, the immune reactions that cause the release of histamines and other immune products will be elevated. This would of course cause the toxins to be produced, where there is a huge possibility of occurring of the bacterial infections, right? So vitamin deficiency would occur, can of course, all of these can contribute towards the increased capillary permeability and prolonged ischemia. If a particular part or the organ of the body is not being supplied with the blood over prolonged time, this can lead to ischemia, which of course increase the capillary permeability as well. The burns of course is one of the example which will increase the capillary permeability, and hence these can cause the edema. The fourth one which we discussed, I mentioned in the previous slide, is the blockage of the lymphatic system, where the lymph won't be able to return back into the blood plasma. So this occurs in the case of cancer, where ascites is the condition which occurs infections in terms of filaria nematodes. This can also occur through surgery. If the surgery has not been performed pretty well. This also includes the congenital absence of if there is an abnormality in the lymphatic vessels, which will of course prevent the flow back towards the plasma from the lymphatic ducts. So edema is also caused by the heart failures as well. In this particular example, the heart of course is not functioning properly, so it will fail to pump blood normally into the veins and from the veins into the arteries. Now this would of course increase the venous pressure, because blood is not pumping blood from the veins into the arteries, so venous pressure and the venous capillary pressure will increase, so it will of course increase the capillary filtration. right? So when the capillary filtration will increase, as this would of course be resulting in the decreased atrial pressure, because the heart has failed to pump the blood from the venous system into the atrial system. So this would of course cause the decreased excretion of salt and water by the kidneys, because the blood is not being transported to the kidney as a result of decreased atrial pressure. So blood volume will further increase slowly and steadily, and when the blood volume will increase, it will of course increase the capillary filtration, and the hydrostatic pressure will increase more. It will of course cause the edema, and if the condition persists, it will make the condition much more severe. So the necessary actions are needed to be taken at that particular point. So there would come a particular point where the blood flow to the kidney would be diminished, and the kidney of course will secrete the renin hormone. And now this particular hormone plays a very important function. So we discussed in the previous lectures as well what are the different functions of the renin, how renin would of course be stimulated in terms of the aldosterone hormone, what are the different functions of the aldosterones. Of course renin, after the secretion of the renin, it would cause the increased formation of the angiotensin 2 and increased secretion of the aldosterone, which of course will be retaining water as well, because the kidney will secrete renin where there is a conditions of less body fluid. So additional salt and water reduction by the kidneys would be taking place. So to summarize, edema caused by the decreased kidney excretion of the salt and water, because salts are being retained. So with the salts, water is being retained as well. So it would of course cause much more swelling in the body. Edema of course will be caused by the decreased plasma protein. And we discussed different examples of the capillary filtration, loss of proteins in the nephrotic syndrome. We discussed the cirrhosis of the liver, where there is a failure of the protein production. So loss of there is less proteins. This would of course cause the edema to prevail. What are the safety factors that normally prevent the edema? So there is a low compliance of the interstitium, in which the interstitial fluid where pressure is the negative pressure range, is in the negative pressure range, so that there is a prevention of the edema to occur, and there is a chance where the interstitial fluid is not being accumulated in the body spaces. So the second thing is the ability of the lymph flow. In certain circumstances, when there is a possibility of the edema to occur, the lymph flow increases to 10 to 50 folds. It of course washes down the interstitial fluid, specifically the protein concentration. Of course, when the protein concentration will increase, of course, it will cause the movement of the water which has been accumulated in that particular compartment where the proteins were present to ooze out, hence thereby preventing the occurrence of the edema. So this is the most important factor towards the prevention of the establishment of edema. So this particular increased lymph flow act as the safety factor against the edema. What are the different fluids in the potential spaces of the body? So potential space includes the pleural cavity of the lungs, the pericardial activity, the pericardial cavity of the heart, peritoneal cavity of the abdomen, the synovial cavity, and the joint cavity of the joint cavity and the bursae. So it's regularly the fluid is being exchanged between these compartments, and the lymphatic vessels, they drain protein from the potential spaces. They play a very critical function as a safety factor towards the establishment of the edema. So edema fluid in these potential, when the edema is being formed, the first thing that will occur is the accumulation of the edema fluid into these potential spaces, and these potential spaces, when the fluid is being accumulated in this particular condition, this is process known as the effusion. That particular fluid is known as the effusion. So these are the pleural cavities, pleural cavity of the lungs, where this cavity. Under these cavities, the fluid can be accumulated, causing the swelling in this pleural cavity. Same goes for the peritoneal cavity, where if there is some problem with the fluid, if there is a fluid imbalance, or there is a condition, let's take an example of HCC, hepatocellular carcinoma, or the liver cirrhosis, where there is some kind of blockage in the portal venous system. So this would of course cause the accumulation of the fluid into the peritoneal cavity. So there is a catheter that has to put into the peritoneal cavity, so to ooze out all the fluid that has been accumulated in this particular cavity. So in this particular lecture, to summarize, we discussed out from the regulation of the body fluid compartments to the body fluid percentages. We discussed about the osmolar substances in the extracellular, intracellular fluid. We discussed what are the different types of the osmotic environments, and how these osmotic environments are being changed when you add a particular solution which is either isotonic, either hypo or hypertonic. We discussed the conditions under which there is a fluid shift, and we discussed what are the conditions that will give rise to hypernatremia and hyponatremia. We discussed what are the causes that can cause the extracellular edema and the edema caused by the heart failure. And finally, we discussed what are the potential spaces in the body in which the edema can occur by the accumulation of the fluids in the body. So that is all for this particular chapter. Thank you for watching, and for more videos, keep watching sqadia.com.